This lecture, the second part of Part 4, will cover the chemical level of organization in the human body and provide a chemical foundation for the course. In this lecture segment, proteins, nucleic acids, and the high-energy compound adenosine triphosphate will be discussed. Let's begin with proteins. Proteins are also organic molecules and have 7 key functions in the body. The first function protein plays a role in is support. Proteins known as structural proteins support the body's cells, tissues, and organs. Proteins are also involved in movement. Proteins known as contractile proteins contribute to muscular contractions. Proteins are involved in transportation. Proteins known as transport proteins aid in the transportation of materials including lipids, gases, minerals, and hormones. Buffering is another process that proteins are involved in. Proteins help prevent drastic changes in the pH of the body. With regards to metabolic regulation, proteins are involved here, specifically enzymes, and help speed up chemical reactions, thus affecting the metabolism of the body. In coordination and control, proteins known as hormones can exert influence on the activities of cells. And finally, in the function of defense, proteins that are waterproof protect the body from its external environments. Proteins known as antibodies protect the body from pathogens. And proteins known as clotting proteins help control bleeding. Proteins are composed of amino acids. Amino acids are the basic unit or building block of these proteins. There are 20 different amino acids found in the body, each of which contains a central carbon atom connected to a hydrogen, connected to an amino group, connected to a carboxylic acid group, and connected to a side chain denoted R, which varies between the different amino acids. Amino acids are linked together by what are known as peptide bonds. These peptide bonds are formed from a dehydration synthesis between the carboxyl group of one amino acid and the amino group of another amino acid. These peptide bonds can be broken through hydrolysis reactions. So here we have one amino acid and a second amino acid. The carboxylic group of this amino acid will donate an OH, and the amino group of this amino acid will donate a H to form water, which is removed in a dehydration synthesis, resulting in the formation of a peptide bond. This again is what links up the different amino acids, and as the chain grows, they become peptides, and then the peptides will become proteins, long chains of amino acids. Proteins are built with a specific sequence of these amino acids. Based on the sequence, the protein shape or structure is determined from several molecular interactions, including the interactions between the different R groups contributed by each amino acid, hydrogen bonding, and also interaction with neighboring water molecules. The complex three-dimensional structure that results is critical, as protein shape determines its function. This is a key concept that we will see again and again. With severe changes to the temperature, pH, or ionic composition of the surrounding fluids the protein is in, protein structure can break down or change, a process known as denaturization. The denaturization of a protein leads to the inability of the protein to function. So if a protein denatures, it loses its specific shape. And as a result, if it does not have a specific shape, it will not be able to do its specific function, and it no longer will be a useful protein to us. Enzymes are essential proteins in the body that catalyze reactions. In the previous chapter, we saw how enzymes achieve this by lowering the activation energy of reactions. In an enzyme-catalyzed reaction, the reactants are known as substrates. Substrates plus an enzyme yields a product. It is important to note that the enzyme does not become part of the product. Enzymes are not consumed in reactions. In fact, they are free to move on and catalyze other reactions after a reaction is complete. So the enzyme itself actually does not become part of the product, but instead is released and able to be reused in subsequent reactions. Each enzyme has what is known as an active site, a specialized region of the enzyme where the substrate binds. The shape of the protein provides a specific active site that will allow the binding of only specific substrates. This is known as the lock and key model and is what gives enzymes their specificity. Let's examine how an enzyme functions. This example demonstrates how a synthesis reaction can be catalyzed by an enzyme. First, a specific substrates will be able to bind to specific active sites on the enzyme. Note how the substrate shape will fit specifically in the shape of the active site on the enzyme. After binding, the enzyme substrate complex is formed. The enzyme temporarily will change shape, promoting the formation of the product. The formed product detaches from the enzyme, and the enzyme, now free and back to its original form, can go on to catalyze another reaction. To summarize, some important characteristics of enzymes include: their function is to catalyze or speed up reactions. They demonstrate specificity. The specific shape of the active site allows only a specific substrate to bind. In other words, each enzyme catalyzes only one type of reaction. Enzymes require optimal pH and optimal temperatures to function. And enzymes are not consumed in a reaction. They do not become part of the product. Moving on to nucleic acids. Nucleic acids are the next class of organic molecules that will be examined. Nucleic acids store and process information inside of cells. A nucleotide is the building block of nucleic acids and is composed of a sugar, either deoxyribose or ribose, a phosphate group, and a nitrogenous base, either adenine, guanine, cytosine, thymine, or uracil. The structures for each of these can be seen in the diagram to the right. There are two classes of nucleic acids, DNA and RNA. It is important to note that thymine is found only in DNA and uracil only in RNA. RNA is composed of a single chain of nucleotides and plays a role in protein synthesis. DNA, on the other hand, is composed of two chains of nucleotides that twist to form a structure known as a double helix. DNA contains the genetic information. The chains of DNA are linked together through weak hydrogen bonding between the complementary base pairs. Adenine pairs with thymine, and cytosine pairs with guanine in DNA. The final molecule that will be examined is adenosine triphosphate, or ATP. ATP is the most important energy transfer compound found in the human body. Adenosine monophosphate is a nucleotide with the components adenine, ribose, and a phosphate group, and this unit as a whole is often abbreviated AMP, again, adenosine monophosphate. Adenosine diphosphate, or ADP, is an adenosine monophosphate plus another phosphate group added on. So here we see our adenosine monophosphate will include this structure here. If we go ahead and add on another phosphate group, it becomes adenosine diphosphate, ADP. If one more phosphate group is added, then we will go ahead and form that high energy compound, adenosine triphosphate. 1, 2, 3 phosphates, hence its name, adenosine triphosphate. The bonds between the phosphate groups, shown here in red, are known as high energy bonds. These covalent bonds store a lot of energy. A large amount of that potential energy that's stored there can be released when one of these bonds is broken. The energy that is released then provides energy that the cell can use to do work. Here we see a molecule of adenosine triphosphate. If a high-energy bond is broken, a phosphate molecule will be released along with energy that can be used for those cellular processes. The ATP then converts to adenosine diphosphate. So here we start with our ATP. We'll go ahead, we'll cleave one of the high-energy bonds, releasing the phosphate and also releasing the energy for our cellular activities. Now we have formed adenosine diphosphate. When energy from cellular catabolism is present in the cell, that energy can go ahead and be stored by taking an ADP, adding that phosphate, and storing that energy in a high energy bond between the two phosphate molecules. So again, we can go ahead and either store energy in those bonds by making them, or break those bonds to release that energy for cellular processes. To review, the major organic compounds of the human body are summarized in this chart. Carbohydrates provide an energy source for the body and are composed of monosaccharides as their building blocks. Lipids play a role in cell structure and act as energy reserves. Triglycerides are the most common lipid and are composed of 3 fatty acid chains and a glycerol molecule. Proteins, as we discussed today, have many functions. They are critical to the function of the body, and they are composed of amino acids. Nucleic acids store and process information and are composed of nucleotides. And finally, adenosine triphosphate, or ATP, is an important high-energy compound in the body. Energy is stored in the high-energy bonds between the phosphate molecules, and this energy can be released when the bonds break to provide energy for cellular processes. The different combinations of lipid proteins and some carbohydrates form structures that work together and build the components of cells. Each cell is a miniature organism composed of these structures built by the biochemical building blocks that were just reviewed. Cells will be discussed in greater detail in the next series of lectures. Let's finish by reviewing with a checkpoint. When 2 monosaccharides undergo a dehydration synthesis reaction, which type of molecule is formed? Also, can you describe in your own words the following: lipids, proteins, and nucleic acids? Feel free to pause the lecture while you formulate your response. When 2 monosaccharides undergo a dehydration synthesis, a disaccharide is formed. Lipids are organic compounds, including fatty acids, triglycerides, steroids, and phospholipids. Proteins are organic compounds that have many functions and are built from amino acids. Nucleic acids are also organic compounds made of nucleotides that compose the genetic material in the cells and also play a role in the regulation of protein synthesis.